Molecular diagnosis. Molecular diagnosis. We know that for effective treatment of a disease, early diagnosis is early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis like serum and urine analysis, that blood tests, they're all said that conventional diagnostic methods here. That early detection is not possible in those cases. Okay. Now, the recombinant DNA technology, polymerase chain reactions, recombinant DNA technology, polymerase chain reaction, we use it to say PCR, that polymerase chain reaction, some of the techniques that help early in diagnosis. Nowadays, you may see that RT-PCR is a very famous nowadays. RT-PCR, real-time PCR test, is very, very accurate test. It was recognized by the government that worldwide that to identify the coronavirus, isn't it? So that means applications of the biotechnology is giving a lot of fruits for us in the for humankind. So some of them, as we are going to discuss here, presence of a pathogen. It may be either bacteria or virus. It is normally suspected only when the pathogen has produced a disease symptom in general. So now we can take example virus, coronavirus. When it gives out as a symptom only, we can recognize we got affected over. By this time, the concentration of the pathogen is already very high in the body. So when you that the time of recognition means that symptoms are exposed means maximum that particular virus is spreaded all over the body. Okay, so that's the problem actually. Then, however, very low concentration when they are in less number, there is no symptom. When they multiply, in large number, when they multiply, the virus is multiplied in the crores, person will get symptoms. Okay, then when we get symptoms, we came to understand actually we have got disease, but almost all it is going to be out of our hands when symptoms are exposed over. So problem, I think you understand what it is exactly, that where the biotechnology we are used that highly useful for us. So when it is in low concentration of bacteria or viruses, at a time when symptoms of the disease are not yet visible, can be detected by. Then here, what is our motto? When they are in less in number even, can we recognize them? Yes, we can recognize by amplification of their nucleic acid by PCR, polymerase chain reaction. So I think you learned the PCR, denaturation, annealing, and extension, the three steps in the PCR, which make the multiplication of the DNA material. Recall that in that PCR technique, we use it to say 30 to 40 automated recycling, that within short seconds of time, that 1 will become 2, 2 will become 4, 4 become 8, 16, 32, 64, 128. Likewise, it will go on, isn't it? So that what we are doing, we are taking the sample from the patient. So that this is the condition. This is maximum when they multiply. But early diagnosis means what we do here. So we'll take the sample from the patient. When we take, we got less number of viruses. When we take the sample. What we do here, by using the PCR, we multiply that particular viral RNA. We multiply in laboratory. Okay, so in laboratory we are multiplying, not in the body. So when we multiply, if two are there, imagine that will be in PCR, we'll make them in the crores in PCR. So then we can identify that, we can detect now. Okay, if there is zero, when even you use PCR, no problem. PCR basically based upon less number may be present in the sample, but by using PCR, we can multiply them, we can multiply. So when we multiply, definitely they are easily can be detected over, isn't it? So that's the technique we use in PCR. Nowadays, PCR, knowing PCR is even good for us. What we do in PCR, that you might have read over newspapers, in YouTube, in various places, PCR usage. Now, RT-PCR, we use it to say for coronavirus. So by this time, let's read it. By this time, the concentration of the pathogen is already very high. Okay, so problem in the Low concentration, no symptoms. When it is high concentration, when symptom comes, what we do? We have to fight a lot here to protect from that particular disease. So how does the PCR is going to be helpful for that? The low concentration of bacteria or viruses at a time when the symptoms of the disease are not yet visible, it can be detected by amplification of their nucleic acid by PCR, polymerase chain reaction. PCR help to detect very low amounts of DNA. PCR helps to detect very low amounts of DNA by multiplication of the small DNA fragment. Particularly, it is very good for virus. The PCR now routinely used to detect HIV. I think we have to add one more, that is Corona, COVID-19. In suspected cases, it is being used to detect mutations in suspected cancer patients too. There even less number of cells are present. We are multiplying them in that way. So it is powerful technique to identify many other genetic disorders such as hemophilia, phenylketonuria, alkaptonuria, that genetic disorders, sickle cell anemia likewise. A single-stranded DNA or RNA tagged with radioactive molecule. What we do here, a single-stranded DNA or RNA is tagged with a radioactive molecule which we call probe, is allowed to hybridize with its complementary DNA in a clone of cells followed by its detection using autoradiography. PCR is just for multiplication. That during that general multiplication also we use PCR. But particularly in these kind of piece that test, what we do, we make that you might know that in case of PCR, the DNA double strand will be separated. Then after we allow the laboratory nucleic materials to be hybridized with them. Okay, so here these particular the DNA complementary strands we are using radioactive probes. Radioactive molecule is allowed to hybridize with its complementary DNA in a clone of cells followed by detection using autoradiography. We use it to detect them. The clone having the mutated gene will not appear on the photographic film because their probe does not show complementary with the mutated gene. Okay, molecular diagnosis is constantly translating new discoveries and novel technologies into useful clinical tests what that detect molecular fingerprints of the disease. You might have learned over PCR, but just for formality, I am showing PCR animation to you that will you can recollect recall that PCR technique. It's a technique actually used to make multiple copies of the segment. PCR is a technique which makes multiple copies of the segment. Let's see from the beginning. It is a technique used to make multiple copies of a segment of DNA of interest, generating a large amount of copies from a small initial sample. Amplification of the DNA segments makes possible the detection of pathogenic viruses or bacteria, identification of the individuals, DNA fingerprinting like, and several scientific research involving in DNA manipulation. We are going to do the same in case of a DNA fingerprinting forensic analysis in human genome project when we learn. This here is most times mentioned in our text. So we will take the sample and we collect the DNA from the sample, and the target DNA will be multiplied in the PCR copies of the target DNA. So that we use in forensic analysis and we use in anthropogenic evolution, that anthropological evolution, that kind of thing. A thermal recycler, that PCR, it carries out the temperature cycles that we need for this thermal recycler. The PCR machine on the screen. So from the DNA, the target sample is taken over. How we'll take, we know that restriction endonucleases will be much helpful for us to cut that. Okay, so then technique, we require some nucleotide free bases required, nitrogenous bases required in the laboratory, and the enzyme, DNA polymerase enzyme is required for extension process. Putting in the machine, automated recycler. It will do. We don't need to go any involvement. It will do itself. In the process, the first, the target DNA is cut down. Target DNA cycle 1, we are now here. You should learn cycle 1. Once you learn the cycle 1, the remaining cycles are going to be the same. The first process, denaturation. We use it to say denaturation. What we do in denaturation, that the double-stranded DNA is going to be cut down in the form of single-stranded under 95 degrees centigrade temperature. The double-stranded DNA will become two single-stranded. Okay. So next we introduce the DNA polymerase enzyme and DNA primers. Okay, so these are the laboratory requirement. DNA primers we require, and nucleotide bases also we require. Okay. So annealing. What is annealing? The primers whatever we introduce, they go into and find their complementary sequences. They'll bind. Okay. The next extension, which is done by DNA polymerase enzyme, will go and attach there, and required nucleotide bases will be collected over. DNA polymerase will collect them and make that particular DNA into single stranded. Now became again the double stranded in both sides. One cycle is completed. One DNA became one DNA. In the beginning, we have one DNA, isn't it? Okay, let me show you that this one DNA, now it became two. Okay, so it became two, like this. One became two. Next, cycle two. Again, denaturation. Denaturation at two double-stranded became two single-strand. DNA primers are introduced, and will they go and combine with a complementary sequence. Annealing is over. Extension by DNA polymerase enzyme. Now it became four. One, two, three, four. Cycle two. Cycle three, you can imagine what happened. Cycle three, four become eight. Denaturation. Double stranded to become single strand. Annealing. Sudden cooling down of temperature we call annealing. Actually. Then DNA polymerase attached extension. Now that it was now again one became two, two became four, four became eight. That's what I said it as 30 to 40 automated recycler. We are multiplied them. We multiplied them. Okay. So that's it. The target DNA, the denaturation, annealing, and extension. We did here. Okay. So that's it. This is about the polymerase chain reaction. In various ways, we use this for to multiply a certain gene which is present in the ancient man in DNA fingerprinting cases to make the copies. So how do we use in our case? That means molecular diagnosis, RT-PCR, real-time PCR, coronavirus. What we do? We do hybridization. That is the only thing we added here. Hybridization. Radioactive labeled DNA probe is going to be introduced over. So that means in every copy after extension, we'll find that the host cell DNA, that DNA of host cell or virus, whatever, and other is going to be radioactive DNA probe. So here hybridization means like this. The extension with radioactive DNA probe. So when we do like that, these particular radioactive probes which are going to be increased over, that is will be recorded on the radiograph. So the PCR is a general technique. We are not going to say PCR is only meant for corona case or only meant for HIV detection. That PCR is a particular procedure which includes the three steps: denaturation, annealing, and extension. We are going to use in various ways. Okay, so do remember this, the sequence also in case of PCR. Denaturation, annealing, and extension. Okay, so that's it about the polymerase chain reaction and their usages. We discussed it now. That their usages are also discovered now that we discussed. So that's it about